Hey everyone, welcome to the first session of our recorded presentations for General Biology 1, BSC1010C. As this is the first session, and this is my first attempt at doing these sessions, it's likely to be a little rough around the edges, but hopefully it will be adequate to what you need in order to explain the presentations. Along the way, we're going to stop periodically. Some of these presentations are going to be somewhat long. They could be several hours long, and that is way too long for you to stay on a single task. You need to break it up into smaller chunks. So at various points along the way, we will stop, and I've included some concept check questions. Now, these are questions that you can think about. We'll stop for about a minute if you want to go on to pause and take a break, get up, stretch, walk around, go and take a bathroom break, whatever it is that suits you. Maybe even turn it off for a while and come back again. It doesn't matter. I'll still be here when you come back. But I will only stop for about a minute, and then we will go on and I will share the answer to the concept check question with you. And so that will break the presentations up into smaller chunks that perhaps will be a little bit easier for you to digest along the way. Certainly will give you some very convenient stopping points. So we'll see how this goes. This is always an experiment, and we'll see if it works. And if it doesn't work, be sure to let me know. Let me know what you think about these presentations. And if they work for you, that's great. If they're not working for you, don't just say they're not working, because that doesn't tell me what I can do to make a change to the presentation. Let me know what you think would be some good additions or some changes that might make this work even better as a good learning experience, because ultimately that's what these are intended to be. They have to be a really good learning experience. So in this session, we are looking at Chapter 1, and here we're going to identify some common themes that we recognize in modern biology that help us understand a lot of the amazing things that we are learning today on the basis of our scientific examination of life. And we're really going to begin that by trying to figure out what we mean by life. That's actually a very, very difficult question to answer. I don't know whether you've even bothered to try and answer that question for yourself. Maybe you want to stop for a minute and think about that one, because it is a very, very difficult question to answer, and I'll explain why in a moment. And then we're going to talk a little bit about evolutionary theory, because of all the themes that do underpin modern biology, that is the single most important, because it really allows us to understand pretty much everything else that we know about biology. If we come at it from an evolutionary perspective, everything else falls into place very, very nicely. So we do need to discuss that a little bit. And I've got a slightly extended explanation of evolution and natural selection in the presentation than what you'll find in the textbook, with some illustrations that I've pulled from earlier editions of the textbook that have simply not been included in later editions of the textbook. They help us explain and understand how natural selection and the process of evolution actually work. And we'll talk a little bit about that. And then finally, we'll talk about the scientific method. And of course, the lab this week is all about the scientific method. So you're going to get a double exposure to what the scientific method is all about. And so we'll discuss a little bit about the scientific method and the relationship between science and technology, because that's also an important question to answer. So let's begin at the beginning. So what you're going to find in every PowerPoint is a slide right at the very beginning that has listed some of the learning objectives, the major learning objectives. Now, there's a complete listing of all the learning objectives associated with the module, and I would strongly advise you to go and take a look at those. And because I haven't had a lot of time to put in all the learning objectives as we currently use them into the PowerPoints, that's part of the editing for the redesign of the course. Hopefully with some of the later presentations as we move through the semester and I have a little bit more time to work on the presentations and the PowerPoints ahead of time, I'll be able to include a full list of all the learning objectives or learning outcomes for each of the chapters as they are documented in Canvas. I would, however, encourage you to use the ones that are documented in Canvas. They are the most recent learning objectives, and those are the ones that you are expected to base your study upon, because that is what you will be held accountable for during the exams. All of the exam questions will be drawn from those learning objectives. So I will make sure that I cover those as we go through each of the presentations, and we'll let you know what I expect you to know and what you really do need to know based on the learning objectives that will help you get ready for the exam. No, I'm not going to give you the actual answers to the exam questions or identify them as actual answers to the exam questions. No, I'm not going to go that far. But you can be very confident that in amongst all of the goodies that I'm going to give you in each of these presentations, you will find the information that you need to answer the questions on the exams upcoming throughout the semester. That much I promise you. So when we take a look at what we think of when we start thinking about biology and we're confronted with this term biology, what we need to remember is that biology is part of a larger discipline called science. And science is really all about knowing stuff. In fact, the term science comes from a Latin word for knowledge. And so science is really all about knowing about things. And specifically, it's knowing about the natural world, what we can see around us, what we can touch, what we can feel, what we can manipulate. And that includes the rest of the universe as well. Although there's some question as to what constitutes the rest of the universe, as astronomers could probably tell you, but we're not going to get into that because that's not part of this course. But the point here is that science is all about investigating the natural world, the things that are around us, the things that we can touch, taste, feel, manipulate, the things that we can interact with. And so when we start thinking about biology as being part of science, we need to recall and remember that biology is all about studying that part of the natural world that constitutes the living portion. So that includes you, me, and every other living thing on the planet, and anything else that is alive anywhere else in the universe, if we were lucky enough to be able to go ahead and investigate that at some point in the future. But for now, we confine ourselves to our planet. And so then we come to the $64, or if you want, $64 million question, due to inflation. What do we mean by life? And as the note says here, it's really difficult to come up with a one-sentence definition of what we really mean by life. And the reason why is because as soon as we try and create a one-sentence definition of life, we immediately find that we're excluding some of those things that we know are darn well alive, simply because our one-sentence definition is inadequate. And this has been an ongoing problem that all biologists recognize. If you try and pin a biologist down and say, give me a one-sentence definition of life, they'll look at you and say, we just can't do it. It's just not possible. But what is possible is to take advantage of something that we've all been familiar with right from about probably age 1 or 2. And that is the fact that we inherently know what is alive and what is not alive. If you give a young child a piece of wood, they can tell you that's not alive, but you tell them that it came from a tree. They know a tree is alive, but the piece of wood is not. And they know the difference, even if they might not be able to explain it to you. We have this inherent understanding of what is alive and what is not alive. And that is because most living things have some common features. And so if you think about life in terms not of so much what it is, very, very difficult to explain, but by thinking about life in terms of what it does, it becomes very, very easy then to describe something that is alive versus something that is not alive. Now, some of these common ideas or these common themes, these common properties that we're going to be discussing today also apply in some cases to non-living things. but it is the combination of all of these that really allow us to define life by the various things that it does. So perhaps the one thing on this list, and you can see a list of some of the things we're going to look at in this presentation. When you look at this list of things, the one that stands out as being something that you'll find in non-living things is order. Pretty much everything else you're not going to find in non-living things, but you will find them as a feature of living things. So order is something that we do recognize. And in biology and in science in general, we talk about hierarchies of order and organization. And you're going to see several of those this morning as we work our way through the presentation. Then evolutionary adaptation, that's going to be something that we're going to talk about very much when we get to the evolution section and the natural selection section of the presentation. Response to an environment, that comes at several different levels, as I hope you'll be able to recognize as we talk a little bit about it. Regulation kind of also comes out of order, but thinking about order in a slightly different sense than these hierarchical arrangements of organization. Here, when we talk about regulation and order together, we're talking about the fact that life and cells, which are the smallest units of life, are basically bags of water in which chemistry occurs, a series of chemical reactions. And for a cell to maintain its function, to stay alive, those chemical reactions need to occur in a very specific order and to be very tightly regulated as a consequence of that. And that's going to become the subject of part of our investigation when we get to particularly chapter 8. We'll talk a little bit about that. And then in chapter 9 and 10, we'll put all the bits and pieces together when we talk about cellular respiration and photosynthesis. They'll come into more sharply focused when we do those 3 chapters as a group back to back. We talk about energy processing as being a part of life, and that's because being alive, maintaining order, regulating things, growing, reproducing, everything that we think of in terms of what organisms do is work. And if you're doing work, then you're going to be utilizing energy. So in some fashion, you must not only be able to gain energy, but you also must be able to process it in some fashion that you can utilize it for the work that you need to do. And again, that's going to come into very sharp focus in chapters 8, 9, and 10 when we talk about cell respiration and photosynthesis, and we talk about energy manipulation in chapter 8. We talk about thermodynamics and things like that. And then we also need to talk about growth and development, which relates to the notion that cells are the smallest unit of life, and where you're dealing with a multicellular organism like ourselves, even though we may start out as a single cell, something that we refer to as a zygote, we also have to remember that that single cell gave rise to all of the 10+ trillion cells that make up your body, with about 220 different types of cells making up that 10 trillion or so cells in your body. And all of those cells and those different types of cells trace their origin back to that single cell, the zygote, the product of the fusion of an egg and a sperm. So that is something that we also see. And even if we're talking in terms of a single-celled organism, like a bacterium, or some of the microorganisms that we refer to as protists, even though they are made up of single cells, they still show growth and development. And then finally, we come to the most fundamental driving force of all, behind all organisms, the need to reproduce, because without reproduction, there is simply no continuity of a species. And one of the ideas that I often mention to class is a question that I float, or a concept, an idea that I float with students in the classroom. So I'll put this one to you as well. is to think about it in these terms. If the last human baby ever to be born was to be born tonight at midnight, how much longer would humans as a species continue to exist on the planet? And once we understand that the longest recorded, accurately recorded human lifespan is 121 years, then it becomes very obvious that if the last human baby ever to be born was born tonight at midnight, humans as a species would be gone in a little over 120 years. That's it. Our existence as a species would be finished if there was no ongoing reproduction. That's how fragile the existence of any species is, but that also underlines the need for reproduction, whether it's reproduction of single cells for repair and replacement in an organism like ourselves, so those multicellular, or in the case of single-celled organisms, where it's a case of producing more copies of the organism, because every time a cell divides into two, you make two new organisms. And so it's reproduction kind of is, and cell division is kind of very important for them under those circumstances. So these are some common themes that, or ideas that we recognize about organisms on the basis of what they do. And it's the sum total of all of these that really allows us to identify living organisms and separate them out from non-living organisms if we have to do it in a formal, organized fashion. So let's take a look in more detail at a couple of these. So in terms of all of those common properties that we assign to living organisms, we can summarize them into these 5 themes or concepts. So we've got organization, and there are a number of different levels at which we can talk about that. There's information, which goes hand in hand with reproduction and the necessity to pass on genetic information from one generation of cells to the next, from one generation of organisms to the next. There's this whole question of energy processing and how do we take that energy in, and in what form, and how do we manipulate that to our own benefit. Interactions. No organism is an island. You've probably heard that no man is an island. Well, this is also true of organisms. Organisms cannot live independently of each other. They must interact in some fashion. And then we have the whole question of evolution. What does that tell us about the diversity of life, but what does it also tell us about the unity of life? Because there is unity to life as well. All will be revealed as we talk about natural selection and evolution. So in terms of organization, hierarchical levels of organization, there are several different types of hierarchies that we can refer to, and we will refer to 2 of them. As we look at these hierarchies, what becomes really obvious is that each level within the hierarchy has its own unique set of properties. But as we transition from one level to the next level in every hierarchy, what we do notice is that there are new properties that emerge at the next level that were not obvious by looking at the properties at the previous level. This is sometimes summarized in this statement: the whole is greater than the sum of the parts. You've probably heard that before, and it's used a lot. But what does that really mean? What it really means is that if you were to break a complex system down into smaller component parts and examine each of those smaller component parts and understand how they operate, and then bring them back together as a way of trying to understand that original complex thing that you were looking at, you're not really going to appreciate the total beauty of the thing that you pulled apart just by looking at its parts. Because what it is as a unit is more than just what its components suggest. So if you want an everyday example of this, think about your car. You know your car is made up of a number of different systems. You've got the engine and you've got the transmission. you've got the electronics, you've got the entertainment, you've got the navigation, you've got the steering, you've got the wheels, you've got a whole lot of systems that are incorporated into the car. You can take the car apart and you can look at each of those systems. You can understand how the engine works. You can understand how the transmission works. You can understand how the steering works. You can understand how the electronics work, how the navigation system works, and all the other things. And you'll learn a lot about your car, but you won't really know how your car works until you bring all of those together and understand how all of those components interact with each other to give you your car. And your car is more than just the combination of those functions, because it's not just those functions, it's how those functions interact with each other to create new possibilities that really makes your car what it is. This is the notion of emergent properties. And we see the same thing in any hierarchy that we build where life is concerned. So yes, we do use the reductionism approach to understanding complex systems. And we do that in biology and in science as well. We take very complex systems like an organism and we understand how its digestive system works, how its circulatory system works, how its pulmonary system works, how its musculature works, how its nervous system works, how its skeleton works. These are all systems that are found inside our body, but we don't get and while we can get an appreciation of all of those systems, we don't truly understand how our body works until we look at all of those systems in combination and see how they interact with each other and build on each other to create the human body that is more than just the sum of those parts. That's one of the issues that we constantly struggle with in science and particularly in biology, where we are dealing with some of the most complex systems that exist. And that is no more exemplified than trying to understand the human brain, a very, very complex system that we struggle to understand because we're trying to use our own brain to understand how our brain works, which doubles down on the complexity of that understanding. But even if we separate the brain from the rest of our body, we still don't truly understand how our brain runs our body until we see how our brain interacts with all the parts of our body to understand how those interactions occur. Again, we run into this issue. Yes, reductionism is a really nice way to understand a complex system by looking at all the parts that make up that complex system. But at some point, we still need to bring them together and realize that there is more to the story than just the understanding of those parts. It also involves understanding how those parts interact with each other. More things are going on than just what is obvious from understanding the parts. So here is one hierarchy that we can look at. I'm not sure whether I can push this out of the way. I'm not sure. I can't see what you're seeing right now. So I'm not sure whether I can push this out of the way so that it's not impacting on the screen, but we'll work through it. So at the very smallest level of organization, the lowest level of organization, we have molecules that are made up of atoms. So we could actually go one step further here and we could go another step down in the hierarchy and we could look at atoms. Or we could go even further down and we could take a look at the particles that make up atoms. And we will do that in the next chapter. Chapter 2 is all about atoms and molecules. But if we're thinking in terms of life itself, then we start with the molecules. And there are different types of molecules that make up structures inside cells, and specifically inside eukaryotic cells, the type of cells that you and I and many multicellular organisms are made up of, referred to as organelles. The term organelle actually means little organ. And you could kind of think of them as being the internal equivalent in cells of organ systems inside our body. They show that level of specialization. So here's a specialized organelle. You may recognize this. This is the chloroplast, the site of photosynthesis. And it has structural features inside it that allow it to carry out photosynthesis. Then all of these organelles assemble together into a cell. And this is a plant cell. These green discs that you can see inside this plant cell, those are the chloroplasts. So there's probably 30 to 50 chloroplasts inside an average plant cell. But there's a lot more in there other than the chloroplasts. And so we have to now take that into account as well. The fact that there's more going on in a plant cell just other than just photosynthesis. And we'll learn more about that when we get to chapter 6 and onwards into, again, chapters 9 and 10. We'll talk more about the sorts of things that are going on in there. Cells then are assembled together into structural features called tissues, which are combinations of different types of cells. So you might have muscle cells in a body, nerve tissue, and so forth. They're made up of specialized types of cells. In plants, there are only a limited number of tissues, but here you can see the photosynthetic tissue inside a leaf. This is actually tissue called spongy mesophyll, and you can see the air spaces around that make up the spongy nature of the tissue. So you've got the cells and then the air spaces, and these air spaces are connected to these openings called stomata that allow gases to move in and out of the leaf and also allow water to move out. And then their opening is regulated by a pair of cells that surround them. You can see them here, called guard cells. These tissues are then assembled into organs and organ systems. And in this case, because we're focusing on plants, the organ system we're looking at is a leaf. And so a leaf is made up of spongy mesophyll and some other types of mesophyll tissue and other tissues, vascular tissue for transporting water and nutrients and from the soil and for moving the products of photosynthesis, primarily sugars, to other locations in the plant where they're needed. But then as we take a look at that, we find that there are more organs and organ systems. So there's not just leaves, there are branches and there's other tissue. There's wood inside the trunk, there's vascular tissue in the trunk, there's roots at the base of the tree. All of these go together to make up an organism, which is assembled from organs and organ systems. Then if you've got a group of the same type of organism, in this case, we're looking at trees, we're looking at the same type of tree. So say we're looking at all the aspens that are growing in a particular location, we say that we're now looking at the population of aspens. So the group of organisms of the same type living in the same location at the same time represent the population. Then if we've got populations of different types of organisms. So we've got not only the trees, but we've got the animals, we've got the fungi, we've got other things. All of those populations are then grouped together to make up a community. So communities consist of all the populations of all the organisms living in one location at the same time. Those then, the community plus the non-living portion of the location. So the soil, the water, the air, all those parts that are not living, then together with the communities make up an ecosystem. So a community is really the living portion of an ecosystem. So if you're thinking in terms of an ecosystem, you've got both a living and a non-living. The non-living is obviously the air, the soil, sunshine, things like that. And then the communities make up the living portion of the ecosystem. And then all the ecosystems that exist everywhere on the planet make up that part of our planet where life exists, and that's called the biosphere. And it is amazing just how much of the planet actually is considered to be part of the biosphere. It's not just the land, it's underground as well, in the rocks. There are areas where we can look within the rocks and find living organisms. Now they're bacteria, but they nonetheless are living in the rocks, in water pockets in the rocks. And we can go as far as about 5 kilometers deep into the rock, deep into the surface of the earth, and be fairly confident we're going to find bacteria living in pockets of water down there. They grow very slowly. We think they reproduce about once every 1,000 years or so. But they're alive nonetheless, and they are capable of reproducing and doing everything else that an organism does. Then when we look at the major part of the planet, the oceans that occupy about 70% of the Earth's surface, surprisingly, most of the ocean is part of the biosphere as well, because you can go to the deepest part of the ocean, what is referred to as the Marianas Trench. north of the Philippines, and you can find organisms happily surviving 35,000 plus feet deep in the ocean. The Marianas Trench is so deep that you could comfortably drop Mount Everest in there and still have about 6,000 or 7,000 feet of water, over a mile of water, sitting over the top of Mount Everest. That's how deep this spot in the ocean is. And up until around 15 years ago, only 2 people had ever been there. But recently, a few more people have managed to get there, to that deepest part of the ocean. But we find that the oceans, in all parts of the ocean, no matter where we look, is also part of the biosphere. And this is one of the things that is so amazing about life here on our planet, just how versatile it is and just how much of the planet is actually occupied by organisms. Much of it is of bacterial or archaeal, that other type of prokaryotic cell, because they are the most chemically flexible and therefore can utilize the most bizarre types of habitats. But even in some of the less restrictive, less bizarre habitats, you're still going to find a broad diversity of life. Life is so flexible and so adaptable, and that's part of its beauty. So this is one hierarchy that we can look at. We will look at another hierarchy a little bit later. Then I've been stressing that the cell is the smallest unit of life, and there are 2 basic types of cells. There are the cells that represent the bacteria and the archaea, referred to as prokaryotic. The term karyote or karyon refers to nucleus, and so pro could also be replaced by the prefix pre, and that means before the nucleus. These are cells that do not have a nucleus. And we can take that one step further and say that they are not subdivided by internal membranes into smaller compartments. The whole cell is a single compartment. That's a prokaryotic cell. And then there are the eukaryotic cells, and I've started to kind of hint at those when we're talking about the chloroplast. Eukaryotic cells do have smaller compartments, internal compartments called organelles. Most important of which is the nucleus. That's the one that you can actually see with a light microscope. The other organelles are typically too small to see with the light microscope, but the nucleus you can. And that's why the nucleus has always been used as the defining feature to separate eukaryotic cells from prokaryotic cells. And again, the karyote or karyon, referring to the nucleus, the prefix U can be translated as true from Latin. And so this would be cells that have a true nucleus. But we take that definition a little bit further and say that there are multiple internal compartments surrounded by membranes. When we get to chapter 6, we're going to be talking about the various compartments that make up eukaryotic cells, because we're going to spend a lot of time talking about eukaryotic cells in chapter 6. So this is a picture of a eukaryotic cell. This is an animal cell. And you can see some of the features. So the nucleus, the big single dominating feature. Then other organelles, including mitochondria, endoplasmic reticulum, and I believe the brown is peroxisomes or lysosomes, I'm not sure which. It's very hard to tell because of the coloration. It doesn't allow you to see much detail. Then you've got the interior material that all the organelles are embedded in, called cytoplasm, and then around the outside is one feature that is found in all cells along with cytoplasm. So there are a number of universal features that are found in all cells, irrespective of whether they're eukaryotic or prokaryotic. One of those is the cell membrane. Every cell has one, otherwise also known as a plasma membrane. The 2 terms are used interchangeably. And the cytoplasm, though that is also a universal feature. Prokaryotic cells have cytoplasm, prokaryotic cells have a cell membrane. They do lack a nucleus, however. Notice, no nucleus, but there is a region where the DNA is found. That's called a nucleoid. To Yeah, I see the transcript said nucleoid. It's nucleoid. I'm not sure whether it's able to read that properly. Anyway, it is what it is. So you can see that a eukaryotic cell is typically substantially larger than a prokaryotic cell. Although the largest prokaryotic cell, largest bacterial cell, is almost 0.5 millimeter in length and is right on the limit of being visible to the naked eye. It's quite large, but that is very much an exception rather than the rule. Most prokaryotic cells are substantially smaller than eukaryotic cells. Then one of the other things that is a common theme is the fact that all cells use DNA as their genetic material. This is the molecule that, along with protein, constitutes the structural units known as chromosomes. We'll be talking a lot more about those when we talk about cell division in chapters 12 and 13. But we'll talk a little bit about chromosomes here as well. These are the structural features that are passed on from one generation of cells to the next generation of cells, or from one organism to the next generation of organisms. And in fact, it is only the DNA that passes from one generation to the next, carrying the genes and the genetic instructions for making the next generation of organisms. It is really just the genes that pass from one generation to the next. And there's been a number of different interesting ideas that have been proposed about the nature of chromosomes and genes and what they truly represent. Maybe we'll talk a little bit about some of those later in the semester when we talk about chapters 12 and 13. But for now, we'll just keep moving on and talk a bit more in general about the nature of DNA. So one of the things that I've already hinted at, again, is the fact that you and I and every other person and every other animal and plant, anything that's multicellular, is the product of a egg and sperm fusion if they reproduce sexually. And the product of that egg-sperm fusion is called a zygote. And that single zygote then divides into daughter cells, which in turn divide, which in turn divide, which in turn divide, and eventually out of that will emerge you and I as individuals, utilizing the DNA that we received in the form of chromosomes present in the sperm cell from our father and the egg cell from our mother. And again, we'll talk much, much more about the process of inheritance and how that works when we get to chapters 14 and 15. We talk a lot about inheritance and inheritance principles in those two chapters. So most of you are familiar with the notion of DNA as a double helix. You can see the double helix nature of the molecule here. It's actually made up of what are referred to as two polynucleotide strands. So here you can see a single polynucleotide strand made up of what are referred to as nucleotides, designated by the four letters A, T, G, and C. You have no doubt heard of those before. But there are two of these strands in a DNA molecule, and there are specific ways in which the nucleotides interact with each other. So you may have heard that A pairs with T and G pairs with C. So here we have an A in this strand. Over here we would have a T. Where we've got a T here, we would have an A over here. We've got the G here, we would have a C here. Where we have the C here, we would have a G in the matching strand. And we say most correctly that the two strands are complementary to each other. They complement each other. And there's also direction in those polynucleotide strands, in that we can identify two ends. And let me fire up my pencil and tablet so I can draw on this for you. So if we give the designation of one end as 3', at the other end would be a 5 prime. On the complementary strand, it would go from 3 prime on this end to 5 prime on this end. And the way we say this is that the two strands are anti-parallel. Meaning they run in opposite directions, like the two lanes on a two-lane road, they run in opposite directions. That would be how we would think about it. By the way, if you see me looking off over here, that's because I'm looking at my drawing tablet. I've got it over on the side here. It has the slide that I'm looking at over here, but I have a pencil, there it is, a tablet pen that allows me to write on the tablet, on the screen, so that I can make annotations. So I'll be writing on here, and from time to time, I may even actually be typing extra notes onto the slides. So that's something that you'll get from looking at these slides as well. So this is the DNA molecule. This is the molecule of inheritance. And we're going to be looking at this in more detail when we get to chapter 16 and 17. We'll look at its function as a molecule of inheritance, and we'll look at how the information is stored in there. We'll look at how the information is extracted. So just a heads up, the information is actually stored by the sequence of nucleotides. So when we read these letters, we're actually reading genetic information. That's how it's stored. And this basically illustrates how that is done. I didn't realize that they were going to pop this in. I'd forgotten that this was here. So basically here is the DNA, and you can see it's double-stranded. You can see the complementation there. So again, if we put some letters in, let me see which one it's. Yeah, so this would be 3 prime to 5 prime on this side. And so this one would be 5 prime to 3 prime over here. Then this is the messenger RNA. It's actually a copy of this strand that runs 3 prime to 5 prime code. It's actually a copy of this top strand. And then these letters are read off in groups of 3, and each group of 3, referred to as a codon, then represents an amino acid. So each of these, and when we get to chapter 17, you'll learn about the genetic code and how each of these groups of 3, the codons, actually code for a particular amino acid, and you'll learn more about that. So this is how you extract information out of the DNA molecule. So you've got the information stored here in the gene sequence, which is always the sequence of nucleotides running 3 prime to 5 prime. That's copied in the form of a messenger RNA molecule, which is then interpreted by ribosomes to create a polypeptide, which ultimately becomes part of a protein. And it's the proteins that are generally considered to be the end product of a gene. It's not necessarily always true, as we'll discover when we get into chapter 17, but as a general rule, that's correct. So the next thing to talk about is the fact that organisms really do interact with their environment. We can't get away from that. You can't live in splendid isolation from your environment. You must interact with the environment. Because out of your environment and your interaction with other organisms, you're going to extract energy for keeping alive. Remember, being alive is work, and work requires energy. So you're going to have to get that energy from your environment, either by harvesting an energy source in the environment or eating something. I mean, that's one of the reasons why we eat, because the food that we take in represents a source of energy. It also represents a source of raw materials for making our own body. So there's a double reason why we would want to, let me take out these, and realize that this would stay in place. Apparently, I don't need to turn on the annotation system in order to write on the screen, because I didn't have it on earlier. So let me keep it off for now so that I can write on it. And I'll only use the annotation if I need to type some text in. I think that's where I'm going to need it. I've not done this in Zoom before. I've done it with the Canvas conference, but I want to use Zoom because I can keep these recordings in Canvas conference. You can't, unfortunately. So yeah, we have these two basic types of interaction. We need to get energy from the environment in some fashion in order to be able to stay alive. And then we need to make sure that the materials of an organism are always returned. There's a chemical recycling going on. So when we die, organisms, particularly fungi and bacteria, will view us as a food resource, and they'll break things down, but in the process, they're returning all the molecules of our bodies back to the environment to be reused again. So there's this ongoing recycling of nutrients back into a form that it can be utilized by organisms on an ongoing basis. So we're going to see these two types of interactions going on. So here is a diagram that pretty much summarizes this idea. So if you are any organism other than a plant, you're going to have to eat something. You're either going to be eating the plants or you're going to be eating something that ate plants in order to get energy. Plants have a unique feature. They can do photosynthesis, and photosynthesis allows them to capture light energy and use that light energy to create molecules that represent a store of chemical energy. And that chemical energy then becomes available not only to the plants, but becomes available to any organism that eats the plant or eats organisms that are eating the plants. As I mentioned, when anything dies, then the nutrients, the molecules, the large organic molecules are then broken down by fungi, like mushrooms, and by bacteria, and those molecules are returned to the soil, and the plants can then reutilize them again for their own growth. And so we see this chemical recycling of nutrients. We also see a recycling between photosynthesis as a way of getting energy into organic molecules, and a process called cellular respiration, by which organisms release that energy in those molecules produced by the plants to make it available for the work of the organisms. Not only is heat given off, which is how all of the energy that comes into the biosphere eventually releases back into the biosphere again. All the light energy, all the other forms of energy that are captured by organisms eventually is released as heat, an energy form that cannot be utilized by organisms. But we also see a recycling of nutrients, not only the large macromolecules, but also the chemicals that are produced and used in photosynthesis and cellular respiration, the two big chemical pathways that we are going to talk about this semester. And there is a diagram that gives us, again, another way of looking at this. So you and I take in food molecules. We process those, and we process them in the presence of oxygen. We need to take in oxygen in order to carry out cellular respiration, and that allows us to process the food molecules and release some of the energy that's contained in them so that we can use them for cellular work. In the process, we release carbon dioxide back into the atmosphere. That carbon dioxide and water, something else that we also release, you may not realize, but much of the water that you pee out is actually the product of cellular respiration. It's a byproduct of the process. You also breathe water out. It's in your breath as you breathe out. So you are actually giving off carbon dioxide and water. That carbon dioxide and water then become the raw materials used by plants to create the organic molecules that you've just consumed using sunlight energy. So the plants capture sunlight energy and make organic molecules from carbon dioxide, or food molecules from carbon dioxide and water. We then eat that, and in the process release oxygen as a quote unquote waste material or waste product. We then take in those food molecules and the oxygen, process them through cellular respiration. In the process, release carbon dioxide and water, which is then reutilized in photosynthesis. Any waste material from the food we eliminate either through our pee or through our feces, and all of that is then broken down, and all of those nutrients are returned back to the soil for the use by the plants again. So there's this massive recycling process going on. So you can see how organisms are not only interacting with their environment in terms of this recycling, but they're interacting with each other in order to obtain energy and raw materials, whether it be an animal eating plants or eating other animals in order to get energy from them, or it's a plant taking in the carbon dioxide and the water in order to be able to create the food molecules. There's this ongoing interaction and recycling. No organism can live in isolation from another organism. We'll talk about this in much more detail in later chapters again. So when energy transformations are carried out, organisms use chemical reactions to manipulate molecules in such a way that energy can either be stored in the products of the chemical reactions, that will be part of the nature of photosynthesis, or energy can be released by the processing of the chemicals in the chemical reactions. That would be cellular respiration, something like that. Those are the two basic types of chemical reactions that allow energy to be stored, or chemical reactions that allow energy to be released. Either way, it is really important that those chemical reactions be regulated. So no chemical reaction in a cell is ever left to chance. It is regulated by a special group of proteins called enzymes. And those enzymes interact with the molecules that are involved in chemical reactions in such a way that the chemical reactions occur under the conditions that exist in the cell and occur in the specific order required by the cell in order for them to remain alive. There is order here. Those chemical reactions have to be tightly regulated and must occur in an appropriate order in order for cells to stay alive. And enzymes are part of that regulation process. But we can also, in turn, regulate the enzymes, because if enzymes are regulating the chemical reactions, by regulating the enzymes, we have mechanisms for regulating the chemical reactions in more detail. And there are two basic ways of doing this: what is referred to as negative feedback, where the process is down-regulated, meaning it is slowed down or stopped, or positive feedback, where the chemical reaction is upregulated or speeded up to higher levels. Most of the regulation is in the form of negative feedback. This is the most common form of negative feedback, and there are multiple examples of negative feedback that we can look at. And we will look at several examples along the way. There are very few really good examples of positive feedback, but we'll look at a couple, particularly when we get to cell regulation, some examples of positive feedback, because it becomes really important in understanding cellular respiration. So more to come later in the semester. That's chapter 9, by the way. Excuse me. So here's an example of a feedback pathway. So if you have too much, excuse me, too much glucose in your blood, the glucose is the blood sugar, that stimulates the production of insulin, which circulates in the bloodstream and stimulates cells to take up the glucose to reduce the blood sugar level. The glucose starts to go down again. When the glucose level goes down, you then need to stimulate the system to release more blood sugar. And so another molecule called glucagon is released that stimulates the release of the blood sugar. So when the glucose level goes up again, that stimulates the release of insulin that then brings the blood sugar back down again. So you've got this alternating up and down of the glucose depending on which of the two hormones, insulin or glucagon, is present in the bloodstream. Insulin is the one that triggers the uptake of sugar. So when there's too much sugar, insulin triggers the uptake of the sugar and reduces the amount of sugar in the bloodstream. If it gets too low, then glucagon is released to stimulate the release of glucose back into the bloodstream again. And in fact, if the blood sugar level goes too low, the negative feedback would be to stop the release of insulin, plus trigger the release of glucagon. Because if you're constantly releasing insulin, you're going to keep reducing the blood sugar. So you could think, if you don't want to address the glucagon question in this diagram, then simply think about it this way. As the blood sugar level gets lower in response to the presence of insulin, the negative feedback mechanism would be to block the release of insulin to allow the blood sugar level to rise again. But that rise in blood sugar level is usually triggered by the release of a second hormone called glucagon. So, as I said, there will be breaks. We've been going for a little while now. Time for a break. So here's a concept check question. Now remember, at this point, if you need to go and take a break, if you need to step away because you're getting a little zoomed out, and I understand that because it's a very real thing. I get zoomed out a little bit too easily at times because of all the meetings that get called, and they're all Zoom meetings because we can't meet on campus, sort of thing. So if you want to take a break, you can either put this on pause or close it. It doesn't bother me because I'm not going anywhere. And I will never know if you do that. But if you want to follow through with this concept check, let's do the concept check question. So now that we have identified some common themes in modern biology, see if you can identify which theme or themes are illustrated in the following examples. Quills of a porcupine, the multicellular development from a single cell to ourselves, or a hummingbird processing sugar to power its flight. There's at least one theme attached to each of these. There could be more than one theme. So I'll stop for a minute. If you go take a break, put it on pause, come back. I'll be here when you come back. So that's about a minute. Welcome back. Let's take a look and see what we think. So quills of a porcupine. That's actually a protective mechanism for the porcupine. That's actually an evolutionary adaptation. This protects the porcupine so that it doesn't get eaten. So that would be an interaction with another organism, an interaction with an environment. It's a protective measure to protect it from getting eaten by a predator. Multicellular development from a single cell. Well, that would be growth and development, I guess. I mean, we had a diagram showing the fusion of an egg and sperm, giving the zygote, little ball of cells, leading to a person. That's really the developmental pathway that we have all followed. So that would be tied up in that. And you could also include in that the passing of chromosomes from one generation cells to the next through cell division, the passing of chromosomes from one generation of organism to the next through reproduction. And then a hummingbird processing sugar to power flight. Well, obviously that's energy processing right there. That's energy processing. And the fact that the hummingbird is getting sugar from a plant in order to provide it with the energy that it needs in order to be able to do the work of flying. So those would be the concepts that we could, the themes that we could apply there. Then you might think of some other things. You might have thought of some other things that could be associated with this. Remember, these are just ways for you to think about what you've seen up to this point and try and make some connections to what we've been talking about. So let's move on. So one of the things that we immediately recognize when we start taking a look at life is just how diverse it is. There's just a huge diversity of different types of organisms. And one of the things that biologists have struggled with since the mid-1700s with the work originally done by a Swedish biologist by the name of Carl Linnaeus, or Carl Linné, or Carolus Linnaeus. You'll find those names used a lot, some variation of that. Biologists have struggled with ways of trying to group organisms together in some fashion. And here we start to encounter another one of the hierarchies. So part of the hierarchy is the product of Linnaeus's original work. Linnaeus basically sorted all organisms into animals and not animals, which he referred to as plants. So if you weren't an animal, according to Linnaeus, you were a plant. And we see this even today by virtue of the fact that fungi are typically talked about in botany classes, because at one time, fungi were considered to be plants and therefore were legitimately part of a botany class which looks at plants. Today, we recognize that fungi are not plants. They belong in their own group. So those of you who have taken biology in the past may have come across what was referred to as the five kingdom system, which consisted of Monera, the kingdom that included all the bacteria as prokaryotic cells, the kingdom Protista, which was supposed to incorporate all the single-celled eukaryotic organisms like amoebas, algae, paramecium, those sorts of organisms, unicellular eukaryotes, and came to include also multicellular eukaryotes that could not be squeezed into fungi, plants, or animals. So it became pretty much a catch-all for anything that was not a plant or an animal or a fungus. And then fungi, plantae, and animalia, fungus, plants, animals. That system was used for a long time until it was realized that, in fact, bacteria are not the only type of prokaryotic cells. There's a second type of prokaryotic cells referred to as archaea, sometimes referred to as extremophiles, because they take the idea of extreme environments to the very absolute limit in terms of extreme temperatures, extreme salt concentrations, and extreme lack of oxygen. They live in environments that not even bacteria that tolerate extreme environments can tolerate. And that's why these are sometimes referred to as extremophiles. Not all archaea do that, but within archaea, there are these prokaryotic cells that can occupy really, really extreme environmental conditions. Along with that was the simple recognition that you could not really support having a kingdom Protista because it really had organisms that were more than what was originally intended in that kingdom. So out of this grew a new system called the three-domain system. And so the three domains are domain bacteria, all of those prokaryotic organisms that were originally recognized as bacteria. Domain archaea, all of those organisms that we now recognize are prokaryotes, but are not bacteria. And there are some very distinct features that separate bacteria and archaea, or that differentiate bacteria and archaea. Then we have domain eukarya, and within domain eukarya, you have the three big eukaryotic kingdoms, so plantae, fungi, and animalia. And then you have a number of smaller classifications or groups that were derived from what used to be the kingdom Protista. And you'll notice that I'm being a little bit hesitant about talking about this because this is an ongoing area of research, exactly what to do with what used to be called protists or Protista, because we don't really know what to make of them. It's still very, very confusing. If you are intending to take biology 2 in the future, you're going to encounter that confusion because we do talk about the diversity of life, and we address all of the kingdoms and the domains, and we do have to deal with the confusion of the protists as part of that. So if you're intending to do general biology 2 in the future, you're going to have to deal with the diversity of protists along the way. There's nothing we can do about that. And well, we just have to deal with it. But within the domains, as I said, we've got the kingdoms, and then we have the Linnaean hierarchical system, which begins with kingdom, then phylum, class, order, family, genus, and then species. And so the most specific designation, the actual name of an individual species, is given by what is referred to as a binomial name, meaning it's got two parts, and the two parts are made up of the genus and what's referred to as the species epithet. So humans you may know as Homo sapiens. So our genus is genus Homo, and our species epithet is sapiens. Wise man is what it actually refers to, because we think we're wise. At least that's how we named ourselves, at least. So all species that share common features are then grouped together as a genus, or share a common ancestor, to use the proper designation today. So all species that share a common ancestor are grouped into a single genus. All genera, which is the plural of genus, that share a common ancestry are grouped into a family. All families that share a common ancestry are grouped into an order. All orders that share a common ancestry are grouped into a class. All classes that share a common ancestry are grouped into a phyla or phylum, and all phyla that share a common ancestry are grouped into a kingdom. Now you notice I keep using the term common ancestry because under the modern system that relies on the domains and the Linnaean classification system, a valid grouping referred to as a clade or a phylogenetic grouping, phylogenetic meaning a common evolutionary history, is grouped together on the basis of sharing all organisms that are descended from a common ancestor and includes that common ancestor as well. So that's why when we define a genus includes all the species that share a common ancestor and can be traced back to that common ancestor as part of the grouping, and so on up through this hierarchy. So each level out, as we go from right to left, includes all the levels further to the left. So the domain includes all the kingdoms. The kingdom includes all the phyla. The phyla includes all the classes. The class includes all the orders. The order includes all the families. The family includes all the genera. The genus includes all the species. It's a nested tree. So you may have seen this sort of a diagram. That's not going to work. Let's go back. I think I may have to turn on the annotation. So you may have seen, that's not working, is it? There we go. That's what I want. So you may have seen a tree diagram that looks like this. Down the bottom here is the common ancestor of all the organisms that are up on the tree, and each branch that ends, so each point here represents a species, or it could be a series of families, it could be anything. We can create tree diagrams in a number of different ways, but at the base of the tree down here at this in this location is the ancestor of all the others that are on this tree diagram. And so you can draw these tree diagrams for any group, any grouping of organisms that you want to create. This is part of the modern biology, what is referred to as phylogenetics, and is governed by what is referred to as the phylo code. You know, I can see again that the transcription service is struggling with this. It's going to be interesting to see what is how it translates everything as we go. Anyway, so when we start thinking in terms of common features, we recognize that these common features are shared because they are derived from a common ancestor. That's the whole basis of the phylocode, of creating these phylogenetic clades, or what are referred to as monophyletic groupings or monophyletic clades. The idea of that tree diagram is that all the individuals that are on the tree are descended from the common ancestor. And that is the explanation for the common features. So when we start thinking in terms of some common features, here we see what is referred to as a cilium. Cilia are found in humans in our airways, and they're also found on the surface of a small protist called paramecium. And when we do a cross-section through them, we see that they share exactly the same features, what is referred to as a 9+2 structure that you'll see when we do chapter 6. So the presence of cilia on paramecium and in our own airways is a common feature, which means that under the phylocode and the phylogenetics system, which we accept as being the explanation of both the diversity but also the unity, tells us that at some point in the deep, deep past, what we refer to as deep time, all eukaryotic cells, because a paramecium is a eukaryotic cell, and we are composed of eukaryotic cells, all eukaryotic cells trace their origin back to an original eukaryotic ancestral cell that had the ability to make cilia, that had this structural characteristic, the 9+2 structural characteristic, because all eukaryotic cells, whether they be part of a unicellular eukaryotic cell or part of a multicellular organism like ourselves, if they make cilia, they have this common feature. It is a shared common feature as a product of common ancestry. So Charles Darwin and Alfred Russel Wallace. So I want to put in so Alfred Russel Wallace. So Wallace also came up with the idea that natural selection is the mechanism of evolution. And in fact, it was Wallace sending a manuscript to Darwin that prompted Darwin to publish his book. He was very hesitant to do so. Oops, that should be Let me erase this. There we go. So it's actually the Darwin-Wallace theory of evolution. That's how we correctly refer to it these days, as the Darwin-Wallace theory of evolution. So both Wallace and Darwin realized that natural selection, a process that favors characteristics that are beneficial and disfavors characteristics that are not beneficial, that explains both the diversity and the unity of life. The unity is there because if a feature works, it continues to work in all the organisms that have that feature and all the organisms that are descended from the common ancestor where those features continue to work. So if we think in terms of cell respiration, the way you and I do cell respiration versus how a plant does it versus how a bacterium does it is essentially the same. The enzymes are the same, the general processes are the same, and that's because evolution has, or natural selection has favored cell respiration because it works. And the way it evolved in bacteria that first evolved this solution to extracting energy out of molecules, got a benefit from it, and all their descendants got a benefit from it. That includes our own cells as descendants, granted, far removed from the original cells, but nonetheless descendants of those original cells that were able to carry out cell respiration. And because it works, and because messing with it too much would mean that it would stop working, natural selection has favored not only the use of cell respiration, but the use of cell respiration or the passing of cell respiration, one generation to the next in essentially an unchanged state, because it works. No need to fix something if it ain't broken, basically, if you want the truth. So possibly one of the ways that you've seen this is thinking in terms of how you might change the makeup of a population. The first thing that we need to remember is that individuals do not evolve. It's not an individual that evolves. It's the nature of a population that evolves. And we can see that mostly reflected in the physical appearance of individuals in the population, or the physical makeup of the population. What we refer to as the phenotype, and you'll learn more about that again when we do genetics and inheritance. So here are some beetles. They live on a dark background, but you can see that they're not all colored the same. Some are really dark, some are moderately dark, and some of them are really light colored. Well, the light colored ones are going to be really obvious to any predatory bird that's just looking for a meal. And so it will preferentially eat the light colored beetles and leave the dark colored beetles behind because they're harder to see. And each generation of beetles are going to be selectively preyed on by the predatory birds in this fashion. They're going to be able to see the light colored beetles much, much easier than they're going to see the dark colored beetles, which means by and large, it's the dark colored beetles that are going to reproduce and leave behind offspring. And those offspring are likely to be dark like their parents. There may be a few light individuals appear, but they're going to get eliminated by the predators. So over time, the makeup of the population is going to change from a mix of dark and light colored beetles more towards the dark colored beetles, simply because natural selection favors the dark color. Their survival is much higher because they're less likely to be eaten. And because they're less likely to be eaten, they survive to reproduce and they leave behind offspring that look more like them. That, in a nutshell, is what natural selection is all about. It favors some traits, it disfavors other traits. The favored traits increase in frequency in the population. The unfavored traits decrease due to natural selection. So, in essence, what natural selection is doing is filtering or editing, because that is another idea that's been floated. It is changing the nature of the population to reflect those adaptations that are favorable and filtering out those that are not favored. They get eliminated over time. So remember again, it's not individuals that are evolving. Individuals may change over the course of their life, but unless those changes are encoded in the DNA and are passed on to the offspring, they're not going to have any effect. And that was the problem with the evolutionary concept of Jean-Baptiste Lamarck, who proposed that any adaptation that was developed by an organism over its lifetime could be passed on to its offspring. We know that that's not true. It's only those that are encoded in the DNA that will get passed on. So if it gives rise to an adaptation, or if there is an adaptation that can be passed on to the offspring that is favored by natural selection, you're going to find that that adaptation will increase in frequency, while the disfavored adaptations get filtered out through the natural selection process. So this is a flow diagram that basically lays out how this takes place. And you can take a look at this and follow through on this. Remember, these PowerPoints are also available in a non-narrated form or an unnarrated form as a download, so that you can look at these in more detail, and you can see them online, or you can download them to your computer if you wish, and they're available in the resources section. Sorry, not the resources section, they're linked in the task list section right now, but I will put them into the resource section as I mount up these narrated presentations as well. So you can take a look at this and follow the flow diagram through. So what we also notice is that if a population of a particular organism gets split into two for some reason, a river divides them, or a mountain range appears due to geological activity or something like that, those two isolated populations will now accumulate their own unique adaptations as a response to the environments that they find themselves in. And over time, they will become new species. They become reproductively isolated. So new species develop. And they become reproductively isolated. Oops, I want to go back. So once the populations become reproductively isolated from each other, then there's a very real potential that they will develop into new species. And so this is how natural selection not only causes changes in populations, but can also, over time, give rise to new species as they adapt to different environments once they become isolated from each other in this fashion. So one of the most famous examples that people may know about is Darwin's finches. And here are some photographs of some of Darwin's finches. The finches, these are actual finches that Darwin collected on his voyage on the Beagle around the world. These are on display at Cambridge University in Britain, because Darwin was a graduate of Cambridge and he donated his collection of papers and his specimens to the university, and these are in the Natural History Museum on the campus of the university. I've actually seen these, and I have a couple of pictures. This is actually one of my own personal photographs from a visit that I paid to the Natural History Museum that has Darwin's finches on display in collection boxes. And the reason why these finches exist as different species is because they have adapted to different habitats and different food sources as a consequence of that over time. So this is a phylogenetic tree. This is a tree diagram that shows the relationship between all the finches that now exist on the Galapagos Islands, and they are descended from a common ancestor that came from the South American mainland, about 600 miles further to the east. The original finches arrived there, probably blown there by a storm. They found a habitat that they could begin to utilize. Over time, as they adapted to different parts of the environment and to the different islands that make up the Galapagos group, there are about 25 islands in total, and there are about 15 or 16 unique different species of finches scattered across the islands. Some islands have their own unique species of finch. Some of the larger islands have several different species of finch because they have multiple habitats rather than just a single habitat. What is of greater interest as well, perhaps, and you can actually find the videos produced by this couple, is that researchers from Princeton University, Peter and Rosemary Grant, have over the last 25, 30 years been monitoring finches on a tiny little island that makes up part of the Galapagos. And it's small enough that they, every summer, have been able to rig fine netting to capture every finch on the island and take measurements and put rings on the ring tags on the legs of every finch, and every time they catch a finch without a tag, they know this is one of the new offspring of that year, and have kept the records. And over about the last 10 years, they have been able to identify the emergence of a new species of finch as a result of a single large male finch migrating in from another island and starting to mate with some of the females of the existing population and producing offspring that are reproductively isolated from the species of finch that is native to the island, but are able to breed amongst themselves. And they've created a new species of finch as a consequence of this. And you can find the videos produced by the Grants in the study area of mastering biology. Pearson has obtained permission to use those videos, and the Grants have produced 2 videos, and you can find them in the study area. It's quite interesting to look at that. And I'll give you another idea as to how speciation can take place and give rise to a new species of finch. So there is a new species of finch emerging in the Galapagos as we speak, which is kind of amazing because we never thought we'd ever be able to document that in real time. So it is the process of natural selection and evolution that actually accounts for both the unity and the diversity. The unity is because of the characteristics shared with the common ancestor. The diversity comes about because some of the features get modified for the purpose of a new use. Because of a use in a different environment. So we don't have an example in here. I don't have a slide that covers this, but I'll give you an example. So when you think of forelimbs, so our forelimb is our arm, and it has a common makeup. So let me show you here. It's got 1 big bone, 2 big bones, cluster of little bones, and digits. If you think about your leg has the same format. If you think about the front leg of your dog, it has the same basic makeup, but the bones are very different. And while they're the same bones, the forelimb of a dog has a very different feature. Same with a cat. A bird's wing is actually a forelimb, and it has the 1 big bone, 2 big bones, cluster of little bones, digits makeup, but it's been highly modified. A whale's front flipper is derived from a forelimb of a terrestrial ancestor that used to walk on dry ground. And it has the one big bone, two big bones, cluster of little bones, digits. But these bones are very short and very thick because now the flipper has got to push against water, something that is very tough to move against. And so that limb has been highly modified. These are much longer, have lots of little bones in them so that they're extremely flexible to allow the flipper to flex as it gets pulled through the water, but to straighten out and become rigid as it's pushed back against the water to allow the whale to swim. And we could go on and on and on talking about all of these modified limbs. What do they have in common? They're descended from a common ancestor called a terrestrial tetrapod. That's the grouping that we all belong to, terrestrial tetrapods. We are all descended from the original terrestrial tetrapod that had forelimbs with that basic form. One big bone, two big bones, cluster of little bones, digits. But each forelimb is the product of a modification or an adaptation to a different usage. One's adapted for walking, one's adapted for swinging in branches, one's adapted for flying, one's adapted for swimming in water. Natural selection and evolution have taken that basic feature and tinkered with it, modified it, edited it, so it can be used for a new purpose. A little bit different from its original purpose, but nonetheless used for a new purpose. That's the nature of natural selection. That's why we think of it as an editing process. Which brings us to our next break. So again, if you feel the need to go take a bathroom break, get a drink, get up and stretch, now's the time. Put me on hold, walk away, turn me off, or stick around, because in about another minute, we're going to continue on. I'll still be here. So here's a concept check question for you to think about. The term editing is often used as an analogy for the action of natural selection on a population. Explain. That shouldn't be too difficult because that's what we've just been talking about. See you in a minute. So maybe I'll come back a little bit early this time, because this one should be a little bit easier to handle. So why do we think about editing? Because remember, as I was just saying, with an arm, a leg, a wing, a flipper, you're starting with the same basic structure, the one big bone, two big bone, cluster of little bones, digits. And rather than coming up with something completely new, you're modifying it. You're changing its meaning. So one meaning is it's a leg for walking on. One meaning is it's an arm for swinging from a branch. One meaning is it's a wing for flight. One meaning is it's a flipper for swimming. You're changing the meaning of that basic structure and putting it to a different use. I'm not sure that necessarily editing is a good term, but it is a term that the book has used on occasion. I prefer to think of evolution and natural selection as a tinkering process, an experimental process. Keep on tinkering, keep on changing things, seeing what new uses can be made of an existing structure, see if they're beneficial to an organism living in a new environment or utilizing a new habitat or utilizing a new food resource. It's all about tinkering and seeing what works. That's really what natural selection and evolution are all about. So let's move on. Let's take a look at the next section. So when we think about evolution as a process, and I just want to bring this down and make sure that I can read all of this because this is kind of important. The role of observation and the hypothetico-deductive reasoning process. So as I mentioned right back at the beginning, science is all about knowing about stuff. That's what the term is. That's what science really, the term science actually means, or the Latin term that science is derived from. It means to know, a way of knowing, of knowing about the natural world. So it's all about being able to test, observe, and measure stuff, things that you can manipulate, things that you can ask objective questions about, which means there are limits to what science can do. So here are some limits. You cannot address subjective questions. So do you find a particular picture attractive or not? That's a subjective question. That's your own asking about your own personal feelings. That's not objective. If I was to ask a different question, however, how many people find that picture to be attractive? Well, that is an objective question, because I can actually go and ask people, do you find it attractive? And collect numbers based on who does and who doesn't. That's an objective question. Subjective questions, can't answer those. Science can't answer subjective questions. We do not or cannot make moral, aesthetic, or similar types of judgments. We can't tell you what's right or wrong, morally or ethically, because again, that is subjective. May be controversial when explaining events previously considered supernatural. This gets to the whole heart of the evolution versus creation question that has been going on for the last 100 years. Science believes that, and biologists believe that evolution is the best way of explaining what we see in the natural world based on our observations, based on our understanding of how things work, and based on the accumulated evidence that we have in front of us. And every time we take evolution and make predictions on the basis of what evolution tells us, and we go out and we collect data based on those predictions, the data supports the prediction and therefore supports the notion of evolution and natural selection, or to put it more correctly, the theory of natural selection and evolution. But of course, not everyone sees it that way, and that's okay. Science is not necessarily the only way of viewing the world. There are other ways of viewing the world, and we should always be cognizant of that. However, seeing as how this class is a science class, and therefore science looks at the world in a very specific way, and it is a biology class, and biology looks at the world in a very specific way through the lens of evolution, that is the lens that we are going to use this semester. You may not necessarily agree with that, and that's okay. I can't ask you to agree with that. But what I can ask you to do is to be open-minded enough and be flexible enough to be able to look at what we discuss through that lens, because the reality is that lens is the best explanation for what we see in the biological world. And much of what you're going to do in your future biology classes, if you're heading into the world of medicine in some way, all of the learning that you're going to do there is going to be filtered, is going to be viewed through the lens of evolution and natural selection. It is how we as scientists, as biologists, look at the world and understand the way the world and life works. Because everything that we do, every observation that we make, every piece of data that we collect continues to support that. And one of the things that you need to understand, and perhaps you don't, but one of the things that you really need to understand is that of all the scientific ideas, the scientific theories that have been proposed, evolution is the one that has been subjected to the greatest amount of testing and has never failed to meet the test, which is one of the reasons why we are so confident that evolution and the theory of evolution is such a powerful tool and is the best explanation for how life works. So there are a number of different ways in which we can approach science, but what we refer to as the scientific method today traces its origin back to Aristotle. Aristotle proposed that scientific research should be based on observation, and that conclusions should be drawn based on those observations. Now, we are more formalized in the way we do it, but we still look at it through the lens of what Aristotle first proposed. So there are two approaches that are combined together. The first is what is referred to as inductive reasoning. This means that you make a lot of observations, you make a lot of measurements, and you draw a generalized, or you make a generalized statement on the basis of what you have accumulated, that information that you've accumulated. So inductive reasoning is all about reaching a general conclusion on the basis of a lot of accumulated observations, or drawing a general conclusion to explain a lot of accumulated observations or a lot of accumulated data. The hypothetico-deductive idea and deductive reasoning is in fact the opposite of inductive. In inductive reasoning, we take a general statement and apply it to a very specific situation. And this is where the hypothetico-deductive idea comes in terms of developing a hypothesis. So once we've created a generalized conclusion using inductive reasoning based on observation, we then might ask a question on the basis of those observations. When you ask a question, which is what science is all about, asking questions, you need to come up with an answer. And the answer is what is referred to as the hypothesis. It's a statement that attempts to answer the question that you've asked. Now, once we've got that hypothesis in place, our presumptive answer to the question that we're asking, then we need to create a predictive statement. This is where the deductive reasoning comes in. We need to make a predictive statement on the basis that our hypothesis is correct. So let me give you an example of this. We observe that a light bulb has gone out. The question we might ask then is, why is the light bulb out? Our hypothesis could be the light bulb is bad, right? The light bulb burnt out. That's why the light's not working. So the observation is the light is not working. The question we're asking is, why is the light not working? The hypothesis is the light bulb is bad. If we were going to create a predictive statement, we would start by saying, if the light bulb is burnt out, there's the restating the truth of the hypothesis. Remember, our hypothesis was the light bulb is burnt out. So if the hypothesis is true, if the light bulb is burnt out, then, and the then part of the statement describes what we should do to show that the hypothesis was correct. What test are we going to perform to demonstrate that the hypothesis was correct? So in the case of the light bulb, well, if the light bulb is burnt out, replacing it with a working light bulb should result in the light coming back on again. Right? So we could go, if the light bulb is bad, then replacing it with a working light bulb will result in the light coming on. There's a predictive statement. There's our deduction. If light bulbs burn out, so in this case, the light bulb is not, or the lamp is not working because the light bulb is burnt out. So we're drawing a specific conclusion that the light bulb is bad on the basis of a generalized observation that lights go out because the light bulb burns out. Hopefully that makes sense to you. I feel like I'm being a little bit disconnected, but hopefully that makes sense to you. So this is how we go through the scientific process. The inductive reasoning is the first part where we collect observations and reach a general conclusion. On the basis of those observations, we then ask a question about that. That leads us to developing the hypothesis, a potential answer to that question. We then create a predictive statement, which is the deductive part of the process, tying the hypothesis to the predictive statement. That's the hypothetico-deductive statement. That predictive statement describes what test we should be performing, in other words, what experiment we're going to do, and what we expect to see as a result of the experiment should the hypothesis be correct. Then we evaluate the experiment and the hypothesis by looking at the results that we get and seeing whether they match what the predictive statement said, our deduction said. Because if they match the deduction, remember the deduction was drawn on the basis of the hypothesis being correct. So if the results match the prediction, they now support the correctness of the hypothesis. They don't prove the correction of the hypothesis, because in science we don't prove things. We provide supportive statement. We demonstrate support for the hypothesis. Only math has proofs, because the important thing to understand about a proof is it's always correct. It's never incorrect. And in science, that's not the case. I like to say that our understanding in science is only as good as the data we have today. If someone comes with data that causes us to change our view tomorrow, then we have to change our view on the basis of the data. Nothing is unalterably correct in science. However, some things are almost to that point, that we raise them from the level of hypothesis to the level of theory. And that's what we mean by a theory in science. A theory is something as close to an unalterable truth as we can get in science. It doesn't mean that we won't change it. The theory of evolution, as Darwin explained it and Wallace explained it, is somewhat different from the way we would explain it today. But that's because we have more information that allows us to build a more elegant, more detailed, more expansive theory of evolution than Darwin could have imagined. But at the heart of the modern theory of evolution is still the basic ideas of Darwin and Wallace. Those have not been thrown away. Those have not been negated. They are the starting point of a broader, more elegant, more detailed, more complete theory of evolution that we have today. But that's only because we have more data that allows us to expand on the original theory of Darwin and Wallace. And that is also the nature of theories in science. There is nothing to say that they can't be expanded upon and improved upon and developed to be more elegant and more expansive and more of an explanation of what we're seeing. That is not outside the realm of science. That is part of the process. So in terms of a hypothesis, and this is the reason why we struggle with subjective questions and questions of morals and ethics and things like that. A hypothesis, the basis for developing our experiments, our tests, is that it must be testable. In order for it to be testable, it's got to be falsifiable, meaning it has an equal possibility of being true or false. If something is always true, you can't test that, right? If someone is holding on to a hard-held belief, you can't test that belief because for them it's always true. That's why we don't get into questions of morals and ethics and subjective questions, because our tests have to be falsifiable. They have to have an equal opportunity to be true or false. That's it. In terms of the scientific process, there always has to be this possibility of the hypothesis being true or being false. It has to be testable. It can't be testable if it can't be falsified. So here is the next flow diagram that shows you how the scientific process works, and you can again follow this one through and see how how hypotheses are tested on the basis of one, that is hypothesis that is correct, correct and one hypothesis that is incorrect in this flow diagram. The other thing that we should always remember is that whenever we do experiments, there's always going to be a control group. This is a group that is used as the yardstick against which we measure the results that we get from our experimental group. So in most experiments, and this has been true in all the testing of the COVID vaccines that have been out there, that have been tested over the last year, there's always going to be individuals who are going to get the experimental treatment, and there's going to be a group that don't get the experimental treatment included within the experiment. So the control is treated in exactly the same way, or the individuals in the control group are treated in the exact same way as the experimental group, with one exception. They don't get the experimental treatment. That means that when we look at the results from the control group and we look at the results from the experimental group, hypothetically, at least, the only difference is the presence or absence of the experimental treatment, whatever that was. And so whatever difference in the results for the two groups should be the product of that experimental treatment. And we have ways of testing to make sure that that is the case. Sometimes it's not always the case. Sometimes we realize that what we did is not necessarily testing what we thought we were testing. We need to come up with a better way of doing the test. In other words, we need to redesign our experiments. This is, again, one of the reasons why science takes so long. Sometimes we realize the experiments that we're doing are not really testing what we thought we were testing. And we need to redesign the experiments in order to be able to test what we want to test. It is part of the challenge of doing science, and it is a very interesting intellectual exercise. I used to really enjoy research when I was actively involved in it. Just the intellectual challenge of coming up with really good experiments that actually tested what I thought I was testing. And then on those occasions when the experiments didn't give me the results that I was expecting, trying to figure out whether this was a problem with the experimental technique or a problem with the hypothesis. Always part of the puzzle when you do science. So we've talked about, the last couple of times, we talked about theories and how they differ from hypotheses and how they, so when we use the term theory in non-science realms, the term that a scientist would use there is hypothesis. So when you might say, well, it's theoretical, and you're not using it in a scientific fashion, what you really ought to be saying is it's only hypothetical, because hypotheses remain to be tested. Hypotheses need accumulated observations and experimental results to be supportable. A theory goes beyond that. It explains many, many hypotheses. It's an umbrella explanation, and more to the point, it's also can be used as a predictive tool. That you can look at the theory and look at what it says and make predictions about what you should see on the basis of what that theory is predicting, and be able to go out and collect results that are supportive of the prediction and reinforce the theory. And remember, the theory is that umbrella explanation of many, many hypotheses and many, many observations. It is a much bigger umbrella idea than a hypothesis. A hypothesis focuses on a very small single aspect of the subject in question. A theory is a bigger umbrella coverage of all of that and more. So, another break. We're getting very close to the end of the presentation, however. So, if you want to hang around for a couple of minutes, well, in a minute, we'll get through the rest of the slides, but here's another concept check for you to think about. And again, this is something that I've just been talking about, so it shouldn't be very difficult to come up with an answer to this. Why is natural selection called a theory and not a hypothesis? And perhaps we'll just move on from here rather than waiting for too long. Why is it a theory and not a hypothesis? Because it does explain so much of what we see in the natural world. It does give us the explanation for the diversity and the unity of life. It explains all of that, both at the big picture and also in fine detail. It's not open to question. It doesn't require further testing to demonstrate that it is the best explanation for what is going on, because it's been tested so many times, and every time that it's been tested, the results have simply been further reinforcement of the reality of natural selection as a process and as an explanation of the way life works. So that's why it's not considered to be a hypothesis. It truly is a theory. So it's the theory of natural selection, the theory of evolution. So the final thing is to recognize that science does not exist in a vacuum either. It is part of what society does. It is a social activity. No researcher does their work in isolation. They work within research groups. They interact with other research groups. It is a worldwide activity, and research in the same subject is being carried out by different groups located in different areas. As I said before, knowledge is conditional. Our understanding of the way the world works is only as good as the data we have today. If better data comes along tomorrow, then we have to change our view. And we have to be big enough and we have to be flexible enough to acknowledge that the old view is wrong and the new data proposes a better view. Sometimes that's not always the case, and because scientists are human, it's not unknown for scientists to fight over the way information and data should be interpreted, and sometimes it's got pretty heated. I recall a couple of situations that I've been present at where scientists have almost come to physical conflict because of their disagreement over the way things should be interpreted and how personal they were taking things, way too personally probably. But that is the nature of human, that's part of human nature as well, yes. So here is another diagram that shows some of the ways in which science and the scientific process connects to what we are looking at as far as a society, as a culture, the benefits that we might get from it, the way the community might influence, the way science might influence the community, the way science influences the discovery process, and the way the discovery process influences science and influences society and influences communities along the way. You can see that it is very much a very, very highly interconnected process, which is one of the reasons why science is so important and why we should continue to support science, because it has so many benefits to us as a culture, to us as a society, to us as individuals. Because in the end, what we refer to technology is really just science being applied. So yeah, science makes discoveries. That's what basic science is all about, making fundamental discoveries about the way the world works. But those discoveries may not be of value in terms of what you can do anything with them. But think about all the electronics that you use today. Back when IBM first invented the transistor, or IBM scientists first invented the transistor, no one had a clue what to do with the transistor at all. It was a vacuum-filled glass tube with pieces of wire in it that showed a very interesting effect as a consequence of passing an electric current through some of the wires. But no one knew what you could do with it. But today, we pack literally billions of transistors in a different format, granted, but they're still just transistors, onto a computer chip. And those computer chips are at the heart of the computer that you're using, or the cell phone that you're using to watch this, the TV that you've got sitting in your room, the cell phone, all of the electronics that you are dependent upon, the electronics in your car. One of the issues right now is that there are not enough computer chips for the car manufacturers to make cars, because cars are massive computer chips these days. Aircraft are massive computer chips. They're flying computers. Your car is a mobile computer. Today, we could not imagine our world without transistors, but that's the application of the discovery science of that original transistor by the IBM scientists, and finding a way to utilize those transistors. That's the technology. That's the connection between science and technology, but that's also the difference between science and technology. Science makes the basic discoveries. Technology is about finding ways to utilize those discoveries in ways that are practical. So science has benefits. It's not only because it's a way of finding out about how things work, about the nature of the world around us. It can also have very remarkable applications once someone figures out how to use it. And that is something that we see in biology as well. And we'll see some of that this semester as we work our way through the chapters that we have to look at this semester. So I think I've got one more concept check. Yeah, so the final concept check, explain the difference between science and technology. I think I've just done that. So we probably don't need to talk about this very much anymore, but that's it for this chapter. We are done. The first of 17 chapters in total. I will be working in the next couple of days to make sure that we have the presentations for the rest of this week and moving on into next week. Hopefully I'll be able to work on the presentations for next week later this week after I get all the presentations for this week done. And then we'll be good to go, and you should find the presentations available day one of every week, and there won't be a delay. As always, if you have any comments, concerns, or questions regarding anything that I've discussed in this presentation, please come and see me during office hours. My office hours are posted in Canvas, they're in the syllabus, and you should feel free to come and see me in office hours here in Zoom at the times that they are posted. If you want to book some time, I am going to create booking times in the Canvas calendar. You'll get a half-hour block that you can put aside. You'll also get notification of availability of time blocks so that you can book a time when you're available and when no one else has got time booked, and that time will be yours. Otherwise, if you don't want to book time, you just want to take a chance and come and see me, by all means feel free to do so. So that's it for this presentation. I'll see you again when we cover the next one.